Hi, this is Hillary, and I'm presenting my hair pulling concept for Chapter 4. I chose the basic structure of the bacteria cell wall. I'll go over some features and then I'll wrap up with some terminology. So bacteria are most commonly either gram-positive or gram-negative. Gram-positive cells are going to be different from gram-negative cells. And it's the distinguishing characteristics in the cell wall of bacteria that's going to indicate whether the bacteria is gram-positive or gram-negative. Most important features of the gram-positive cell are the thick layer of peptidoglycan, so that's kind of drawn here, and also the phospholipid membrane. You can see that drawn here. It's a lipid bilayer. The peptidoglycan layer helps the bacteria maintain its shape and it provides structure. So there's also an inner wall zone between the thick peptidoglycan layer and the single membrane. Two main components of the plasma membrane are the lipid bilayer, and then there are also different types of proteins that are found in the plasma membrane. Specifically, lipoteichoic acids. These anchor through the peptidoglycan layer onto the lipids that are in the lipid bilayer of the plasma membrane. So that's kind of sketched here. The teichoic acids anchor the cell wall to the plasma membrane again. This is, so you could think of the teichoic acid as an attachment protein. It also contributes to an acidic charge on the outside of the cell. So that's a general overview of the gram-positive cell wall. Let's take a look at the gram-negative cell wall. The gram-negative cell also has a plasma membrane, and this plasma membrane is also composed of a lipid bilayer that contains proteins. It also has a layer of peptidoglycan, but compared to gram-positive cells, this layer is much thinner. Another difference is that the peptidoglycan layer is anchored to an outer membrane. So here's your peptidoglycan layer, and then you can see the outer membrane here, or phospholipid bilayer. The outer lipid bilayer contains proteins called porins. So those are going to be found in this layer. The porins allow for passive diffusion of things from the outside into the space between the two membranes. So you could think of that, that's actually called the periplasmic space. And I've drawn that a little bit here, these spaces between both layers there. Another important feature of gram-negative cell walls is what's on the very outside of the outer membrane. LPS, or lipopolysaccharides. This is a component that's only found on the outside of a gram-negative cell wall. It can also be referred to as endotoxin, lipopolysaccharide. It's a big word. So again, kind of some important terminology for the cell wall, either gram-negative or gram-positive. We've got peptidoglycan, which is the protective layer, a protective layer for bacteria. It's composed of amino acids and glucose, proteins and sugars, so two molecules in peptidoglycan, acetylglucosamine, or NAG, acetylmuramic acid, or NAM. So in order to combine these two molecules together, there's an enzyme called transpeptidase. Trans meaning across, so these are cross-linked, and this forms the layer of peptidoglycan in gram-positive and gram-negative cells. So because it's cross-linked, it's a strong layer of protection. Lipoteichoic acid, again, it's only found in gram-positive cells, and it provides cell wall rigidity. It has also antigenic properties, so the ability to stimulate an immune response in our bodies. For example, like a fever. It recruits leukocytes when the body detects an invader, so that's pretty cool. And then also we talked a bit about periplasm, periplasmic space. It's in both gram-positive and gram-negative periplasm, but it's more obvious in gram-negative than it is in gram-positive. And then also another key term we talked about was porins. So these are found in the outer membrane of gram-negative cells. These are like little channels that allow things to come into the membrane and other things not to come in. So for example, bacteria love glucose, so this would be allowed to come in. What wouldn't a cell or a bacteria cell want in that, want to be allowed to come in? Things like antibiotics. So some bacteria have gotten resistant and have learned how to block out antibiotics, which come in through the porin and kill the bacteria. So this advancement would keep the bacteria from being susceptible to being killed by certain drugs. Penicillin prevents peptidoglycan from forming. So that's an example of an antibiotic. It makes the cell wall useless and vulnerable to rupturing or lysis in gram-positive cells. In gram-negative, there is a cell wall and a thinner second membrane. Or I'm sorry, in gram-negative, there is a cell wall, but it's thinner. The peptidoglycan layer is thinner. There's also a second membrane. So the gram-negative cell is not going to be as affected by penicillin. That wraps up some of the key features of the bacteria cell wall. Thanks for listening.